The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar, Biocompatibility as a Critical Design Input. We'd just like to cover a few housekeeping items before we get started. If you missed one of our webinars or would simply like to refer to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can also receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following us on LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize risk. So let's go ahead and get started. I'd like to introduce you to our speaker today, Dr. Helen Ragel, Senior Biocompatibility Expert. Dr. Ragel is a globally recognized expert in medical device biocompatibility. She has extensive expertise in writing biological evaluations and assessments for regulatory submissions. These documents focus on using a risk-based approach in defining the path to evaluating medical devices with the US FDA and EU regulatory bodies as the primary audience. She works closely with medical device manufacturers and global regulatory bodies on a daily basis. Thanks to her distinguished academic background with over 10 years of experience in scientific research, she has a broad knowledge of biological systems and the analytical procedures used to evaluate biocompatibility in vitro and in vivo. She also is an active member of ISO and ASTM working groups related to biological safety evaluations. So now, beginning today's presentation, I'd like to turn the time over to Dr. Helen Ragel.
Thanks, Mike, for the very nice introduction. And hello, everyone. Welcome to this webinar where I talk about the most awesome and the greatest subject of all, biocompatibility. And today we will focus on biocompatibility as a critical design input. Now, first, I wanted to give you a little overview of the talk itself. We'll first dive into what is biocompatibility and why do we even talk about it and why does it matter. And then we will go into some sections of the guidance documents, the overarching documents for biocompatibility, ISO 10993-1, as well as the FDA guidance document on the use of 10993-1. Then we'll talk about more about this criticality of starting to think about biocompatibility and when do you need to start thinking about it, and what the guidance says and what we really need to do. And then we will wrap this up with talking about changes, because that is sometimes, I feel like, the crux of this industry is that once you have something on the market, then something changes, and now what do you do? So let's dive in. Let's talk about the big picture first. Why do we care about biocompatibility in the first place, right? What is it, and why do we need to evaluate it for the medical devices that are going to be used on patients? Now, I truly believe that every manufacturer of medical device that is out there, they have a good intention of creating something good for the patient, right? They have these smart people sitting down, they are coming up with strategies and products that would either help to treat the condition or maybe better the life quality of life of these patients. So I think the intent there is always good. Now, I have, from personal experience and from my work experience, seen that good intentions, unfortunately, do not always translate to great outcomes. And, as I said, from my personal experience, I have many of examples from my childhood where I intended to do a really sweet and nice thing, but then in many cases, it did not turn out the way it was intended. And I have to give credit to my mom for being so patient with me. Now, we want to make sure that that doesn't happen in real life, in clinical use of these medical devices, right? Because, I can be scowled at as a child for doing something that wasn't really, the best of plans, but we cannot have that happen with the patients when they need the care the most. So really, we want to make sure that biocompatibility as a term of making sure that these devices that we put out there do not inadvertently cause harm to the patients that we're trying to treat, that is really the key to why we're doing this. Another thing that we need to think about is that we usually look at the devices of how they're going to be used, the intended use of device, but there's often cases where we also need to take a step back and think about what can go wrong with my device. And how do I mitigate those risks? For example, if you have a device and there's some pieces that could maybe during surgery accidentally break off, right? And then what happens if that piece ends up in the patient? Is it okay to leave it in the patient, or do we need to take more drastic measures and remove that part? So that has happened in the past. I just want to make sure that people are thinking about these things. Obviously, we don't want to go into evaluating the moon dust, like we call it, like really improbable situations, but we do need to kind of at least think about it when we're designing our device and looking at biocompatibility. And the last thing that biocompatibility evaluations encompass is what happens if there are changes. Like I mentioned, there's tons of things that are already on the market where we have issues with supply chain. COVID has been the greatest example of the supply chain issues. So now if we're faced with that type of situation, what do we do, and how much do we need to then go back to proving the biocompatibility of the final finished device? I always say, so this is like my favorite slide for biocompatibility. When I was expecting, my husband said, this is the one book that you need to read when you're trying to raise a good kid, right? And so if you haven't read this book, really focuses on knowing your child, right? So knowing what that can they do, what are their ups and downs, and then really trying to hone in on what you need to focus on as the critical points, and then letting the rest of the stuff that doesn't really matter kind of slide, right? So really what I say, we should be taking the same type of approach to biocompatibility evaluations. We need to evaluate the biocompatibility with love and logic, meaning that we need to know our child, we need to know our device that we are making, and what kind of risks does it actually carry? What should we focus on, knowing what we know about how it's manufactured and what it is manufactured from, and then defining the path to demonstrating that when it is used as intended, intended, it is not going to cause harm to the patient. So think about this: know your child, know your product, do the assessment with love and logic. So when we think about biocompatibility, so I oftentimes have people come to me and say, Helen, I have this really cool medical device, and this medical device is meant to be, having the patient live long and prosper. Okay, so I said, okay, that's a great idea. So what I need to know is, how is it going to be used? And really trying to explain, like, I want them to explain to me, how does it contact the patient? I need to know, like, where is it going to be put, how it's going to be used, how frequently, what's the duration of use, and also we want to know if there are any special considerations. For example, if you have a 3D printing process for your device, that carries some additional risks that need to be considered. For example, there could be residuals like particulates from that 3D printing process that are not normally present if you do the manufacturing through and other types of processing, like maybe injection molding or sintering. And another thing that we need to think about is, knowing all of this, is we need to define, based on the intended use, the type and duration of use, as well as the special considerations, what biological risks are there present for this specific device. And more importantly, how are we going to prove the safety of this particular device? Now, before, in the old days, the old school approach was to just kind of test for everything. You look at the table in ISO 10993-1, and then you just say, these are the endpoints to be tested, and you just test them. If the test comes out as clean or passing, then you're good. However, first of all, I do wonder sometimes that if we are just following that strategy, are we really capturing all of the risks that are associated with some device, right? For example, this concern for particulates, right? If we have a medical device that is only used, let's say it's a little syringe that it injects something into the eye, right? And the syringe is super small. Let's say the needle itself is like, the type of needle that you can use for insulin injection. And so the contact is very short. The contact surface area is very short. So the risks there really are very minimum, right? However, I would be worried if there would be particulates in there, because I don't want anybody to have potentially particulates injected in the eye. However, if you look at the 10993-1 as it stands right now, it talks very little, actually not at all, about particulates. So really what I'm trying to say is that we need to have a complete overview of your specific medical device and understanding what are risks, and then defining the strategy to prove. So, and a lot of the times we have also a bunch of available information. Let's say you have a catheter that is already on the market, it's cleared, it's being used. Now you're doing a Gen 2 for that catheter, or maybe an adoption to that product family. We obviously want to make sure that we use all the available information from what you already have before we start to go into more testing. So before, it was not that common to use this approach, but why would we want to use another set of animals, another set of time and resources and money to prove what we already have available? And the second point is that defining what we currently have and what are the risks for this device in general, then we can really define what gaps do we have in the knowledge, and what do we need what type of testing data do we need to close those gaps. So this is the very famous current table A1 in the 10993-1. And here you can see there's lots of different biological endpoints that should be considered given what type of device you're handling or you're producing and trying to prove biocompatibility for. But now the main thing that has been changing is that when we have the table, there's there's lots of E's, right? So that was the big change from the previous version of the 1093-1 that was released in 2009 and to the one that was released in 2018, that all of those X's turned to E's, and the E means that the endpoint really needs to be evaluated in the risk assessment. Rather than just jumping into testing, you need to look at all of the available data, everything that we know about the materials and processing, as well as knowing how your device is used, and then defining, is the endpoint already covered with the available data? Does the endpoint even matter? And what type of testing, if at all, is needed to complete the data set for your given specific device. And then really it says for particular medical devices, there is a possibility that there could be fewer endpoints, but also additional endpoints, depending again on the device type and how it's used, right? For example, some degradable devices, they can degrade really fast. So you can say like, oh, hey, I'm having only a limited contact, right? But if it is degradable, we need to know also where it ends up in the body, right? It might be degraded from the site of injection. However, it could result in the distribution of that substance or some chemicals from that substance that you are injecting in some other part of the body. And there, until it is really, we know what's happening there, we cannot just assume that it's limited contact and it has nothing to do with other biological endpoints that have not been marked as E for that limited contact. So just things to think about. And this risk-based approach really has been going through the industry to really make it so that people stop and think about their products before they start testing. I thought that on September 8th, 2023, it's a beautiful morning where FDA came out with their revised guidance document on the use of 1093-1. And what really changed was this attachment G. So we know that, lots of products that are only used on intact skin, right? There's a ton of things that just contact the skin. Maybe we should be a little bit more lenient on their biocompatibility testing requirements, just because we know so much about these materials, and we know a whole lot from previously cleared devices that they really carry very low risk for biocompatibility. Now, on September 8th, 2023, this became part of their guidance document, the biocompatibility of certain devices in contact with intact skin. So what it really says is that FDA believes that a certain set of materials pose a very low biocompatibility risk based on the history of use of these materials in currently cleared and marketed products. So really what it means is that FDA agrees that intact skin contact can be considered as a low risk profile for biocompatibility, and they propose that instead of providing testing data for these types of devices that fall under this attachment G, that you could use a least burdensome approach and instead of testing data, provide additional quality control measures to prove that and have control of your manufacturing process, the materials that are coming in, and that you still should do a risk assessment to demonstrate that yes, it's low risk for this type of application. However, you don't need to necessarily test these products. And this supports the principles of 3Rs, which means to reduce and replace and refine animal testing, because we really want to make sure that when we are going into testing, we first understand, is it really necessary to do, let's say, sensitization testing or irritation testing on these animals to provide this data that we can get from other places? So when you have a medical device, I always say, and 10.9.3-1 also says it, that you need to start with a plan, a biological evaluation plan that describes your product, your device, its intended use. I always say pictures say more than 1,000 words. So I would really describe your device in the way that somebody who is not in this industry can also follow how your device is used and can already start to think about, oh, what types of risks do I see when this device is going to be used on a patient? We have to make it so that everybody can follow it, because reviewers of all fields, they might have a better grasp on certain fields, but we don't want to expect that every reviewer knows about everything. So please make it so that everybody understands. You can have your grandma check the at least the intended use part of your plan to make sure that she's like, aha, I get it. And then after that, we need to do a deep dive into the available information. So really, it was about demonstrating first with materials and processing information that you know exactly what goes into making your device, and that it's fixed. Really, in my opinion, that first clause of physical and or chemical information for most of the devices out there, it really is demonstrating that you know what's going on, and if there is changes, then you will be notified, and that kind of prompts you to do a risk assessment, and then this biological evaluation process is kind of turned on again. But yeah, really, you need to provide that data on materials and processing to kind of show you have a design freeze, everything is set, fixed. Now we're talking about the final finished form of the device. And as I said, we also need to start with all the available information. So we really want to highlight in that plan, what do we already know about the device that we can leverage perhaps in order to reduce the testing on the final finished device if it's possible. For example, if we have well-known and vetted materials and processing, like for example, we have a tool that is completely made of stainless steel, and it is passivated for a very standard procedure like an ASTM procedure, and there's nothing else being done on that final device, then I would say that data could serve as a basis of doing your risk assessment. Then perhaps say, knowing what you know, additional testing per 10.9.3-1 to cover those endpoints might not even be necessary, just because these processes and materials are already vetted in the industry. Also, as I mentioned, previous testing data on equivalent devices that are manufactured by you, that's very important, manufactured by you. This is the example of going from Gen 1 to Gen 2, that there's maybe slight improvements in what you have done to your previous form of the device. But really, if you can prove that it's equivalent, then I would say that should be the data that you provide to cover the biocompatibility risks. And now the point about manufactured by you is really critical, because I hear a lot of device manufacturers come to me and say, oh yeah, I use this material, and I do injection molding, and it really is like how everybody else does it in the industry or in that section. And it's true that overarchingly they're very similar. However, these minute details about processing aids and cleaning processes that are undertaken, these can totally change the biocompatibility profile of your device. And that's why oftentimes referring to somebody else manufacturing the device similar to yours is not going to be enough. Another very, very good data point to include in this evaluation of available information section is a previous clinical or real world data. So if your product is already on the market, then if you do, let's say, an MDR remediation, absolutely that data, if you have a catheter that is used for limited contact time, obviously we want to make sure that we use the real world data before starting to go, let's say, another panel of hemocompatibility testing, because that real world data is so much more valuable than running another, let's say, thrombogenicity study in 2 dogs, right? We always have to kind of consider this in perspective of what the testing does and how much, like clinical data could have thousands and thousands of patients' data involved. So now, knowing that, and then knowing that the thrombogenicity data is coming from 2 dogs, right? It's really not comparable. And then, as I mentioned, after knowing all of this information, we need to define what are the gaps in the available data versus the potential risks that you have identified for your specific device. And then based on that, we define a testing plan, if needed, to fill in those gaps. I often say when you have a pretty complex device, which I see more and more often with time. These brilliant scientists working on developing these devices, they have come up with some very clever, unique devices. But a lot of the times that makes some of them to be quite difficult to test, or the approach to be used for showing biocompatibility. So I always say, if that's the case for you, I would recommend to share the plan with your regulatory reviewer before commencing, especially some of these long-term, long lead time tests. And this is also mentioned in the FDA guidance document that the center does recommend reaching out and doing a pre-sub to kind of get their take on you're good to go, or wait a second, think about this first. Oftentimes I find that these pre-sub meetings can be very fruitful. Sometimes not so, but oftentimes I do see the value in getting the agency involved early on, because then they also kind of think about it this way. You can put them in the position where they feel like they are a partner for you through this process of submission, getting onto the market, instead of being confronted at the very end with the data, and then they have to say, listen, I think you did the test wrong. So now when we talk about testing, ISO 10993 series methods, there's a bunch of different methods that can be, in bulk, basically categorized as biological tests that follow the 10993-12 sample preparation methods. And then there's the analytical chemistry tests that are covered under ISO 10993-18. And you can see that the biological tests can be categorized as in vitro tests that are done in basically a test tube, or a cell culture dish, or similar way, and in vivo tests that require the use of the animals. Now, I do want to say that right now irritation is still, and there is an in vitro method available that is also highlighted in the specific ISO standard, ISO 10993-23. However, as of now, we know that some regulatory bodies do not accept that approach. So I still categorize it as a potentially, most likely, in vivo method at the moment. Hopefully, we gain some headway there soon. So now there's a ton of different tests, right? But we always need to think about these tests as tools in your toolbox. And these tools will be used to demonstrate the safety of your device, right? Now, talking about the tools, my husband is, again, the greatest person. He has taught me the importance of having the right tool for the job. And I kid you not, every single time we go to Home Depot, we cannot leave without yet buying another tool, right? So, we have a lot of tools in our house. I'm not saying that you need to be so obsessed with tools, but you should know what the tool can do for you. So know your tool, because let's say if you're trying to answer a simple question, let's say you're trying to put up a picture frame, right? So do you want to go directly to a very powerful power tool that can do tons of things, or can you just go to a simpler tool and the method of just hammering a nail into the wall? So it really is, they both can technically be used, but what makes sense for your current task at hand? That's what I'm meaning with this know your tool business. Now, let's go back to when do we need to start to think about biocompatibility, because that's a really important question here. When we look at the guidance documents, with FDA guidance documents, and we all also 10993-1, they all say that biocompatibility testing should be conducted, or evaluation and testing should be conducted on the final finished form of the device. Now, that's always we hear, final finished device. Like every time I talk to the FDA, they say, we don't, clear any materials or whatever. We only think about the final finished device, right? So that echoes in my mind every single day. Now, that's true. So when we look at, because we really want to make sure that the device, as it is going to be exposed to the patient, is going to not cause harm to that patient, right? So that makes sense. Now, when we look at the whole manufacturing process to get to the final finished device, we have materials that we're using, and then we're using the processing, including sterilization, and then we get to that final finished device that the standard says that's what we need to evaluate. But I have to say, please do not make biocompatibility an afterthought. Even though it says we need to do it at the final finished state, the concept of biocompatibility needs to be taken into consideration already before the final finished state has been achieved. Now, that is even mentioned in the ISO 10993-1 as well. It says material selection and risk analysis are integral components of the design process of medical devices, right? So really, it should be a design process criteria already at the material selection part. Now, I wanted to give you an example, kind of like a true story, true crime story type of thing. So one time we had a client come to us, and they had a device that contained ABS. ABS, as we know, relatively low reactivity. I mean, it's even on the FDA list of intact skin contact, no worries, low risk, everything is dandy, right? Now, we had a client come to me. Oh, I have to say, this is a true story, it's a true crime story, right? And this is not something that happened to my elementary school friends, neighbors, grandmas, pastor. It's not. This is actually an example what we dealt with about 5 years ago at Nelson Labs. Okay, so it's a first-hand experience of like, oh my goodness, we need to know what material we have and what we need to know our stuff. Okay, so the sponsor came to us and they had a cauterizing device. And the cauterizing device had ABS on its close to its tip, right? The cauterizing tip. Now, when we did extractions for biological testing, we did stimulate the ablation process because, when you have cauterizing process happening on the patient at the tissue, the cauterizing needs higher temperature, right? So we said, okay, we should mimic this process in the extraction. So we, made the cauterizing tip heat up during that extraction process. And then we performed acute systemic toxicity study. And do you know what happened? Instantly, a mouse died. Okay, acute systemic toxicity means that you injected intravenously the extract into the mouse and it died in like 10 to 30 seconds. So we were like, whoa, what is it that is going on here? Because first of all, we rarely see acute systemic toxicity study fail, right? And especially in this case, where the reaction was so fast, it made us think, wait a second, there must be something happening chemically, and there's only a certain set of chemicals that can really react with blood so quickly. Okay, so long story short, what we turned out, or we discovered within our process of trying to figure out what's going on here, is that ABS that was used when heated up released some cyanide. So you can see why that poor mouse ended the way that it ended. Now, we didn't know this, right? And it really is, not all ABSs do that, but it turned out that that grade of ABS that company was using was actually meant for car bumpers. So please know your child, know your materials, know what they have, what grade they are. You really need to know this in order to make sure that you are not inadvertently causing a situation like this happen. Now, when do we need to start to think about biocompatibility then? I hope that with the past, horror story, I explained to you why we need to start to think about it at the materials level, not at the very end, but we need to start to think about it as a design input for the manufacturers to make sure that we are choosing the right materials, right? For biocompatibility, I totally understand. We also need to make sure that they're right for function, right? But a biocompatibility concern, or at least the thought, needs to be already put in at the materials level. Now, some tips for choosing materials, right? As I said, you need to know your child. You need to know and understand the material. You need to look at the technical data sheet. What can be offered from the supplier of that material, the manufacturer of that resin, with that material that you're getting, right? I mean, there's lots of great materials out there that are widely used. They have lots of very good information on the material already, like previous biocompatibility testing and master files from the suppliers of the materials. That gives you confidence that you're not going to run into that scenario that we just talked about. I do have to kind of make you like at least check what is in the master file, because sometimes the master file for biocompatibility only contains a cytotoxicity test. So please make sure that if you are referring to a master file, know what's in there. Some master files are extensive, like there's tons of different materials, Nuso, Solvay, they provide great upfront information about the materials, and others have less available. But if you ask them, there could be some internal data that they have not publicly made maybe available, but they can provide you as you are purchasing their materials. So please be aware of that. And obviously, previous experience with the material by you, maybe in a different kind of application, that could also lead you to know, okay, is it going to have, you know, potentially sensitizing effects, or is it going to have, you know, irritation or toxicity profile? Like, what type of things are we talking about? So previous experience with the material always helps. Now let's talk about changes. We all know that there could be supply chain issues. Yes, it has happened many of times when I get called up, Helen, I have this problem. They said they can't provide me this thing. The next batch can come in like a year. And you're like, okay, let's see what we can do. Then there's also alternate sources that a lot of manufacturers use to cover that, you know, that scenario where they run into supply chain issues. So you want to qualify 2 sources of different materials, right? So that makes sense from a manufacturing side, but you also need to make sure that you know that both sources provide you a biocompatible material. And then obviously we also deal with upstream notifications of changes, like for example, there's a discontinuation of use, or there have been some changes in some raw materials that go into the manufacturing of that specific resin. So now what do we do about that? Clearly, the main question from my perspective would be, how do these changes impact the biocompatibility? And again, biocompatibility in the end on the final finished device that is going to be exposed to the patient. So how many times can you think, have I heard this thing that the people say, well, but silicone is silicone, right? And I always have to say back, it's like, yes, it sounds the same, but different types of materials, even though they're marketed as silicone, it really doesn't mean that, you know, you can put that into whatever device you have. Material that may be suitable for one situation may not be suitable for another situation, right? For example, if you have a silicone that you put on to make your bathroom, you know, leak-proof, you don't really want to use that for an implant, right? So that's really the point. You need to know and choose your materials wisely. When you have a change, what I really recommend at first is to compare the specs or the available information to the current material you're using, right? For example, if you have a limited contact device and it uses one type of, I don't know, PBAX, right? So it uses a PBAX from this supplier. And it's, you know, USP class 6, and it's used, you know, throughout your products. And now they're saying, oh, they cannot give you this PBAX anymore. So now you have to look for an alternative. So now you're looking for another supplier for PBAX, but their manufacturing process might be different. However, they might already have available information. It's also USP class 6 tested. It's also widely used in other currently marketed devices. So in those cases, when you can compare the specs of the 2 materials and they, for the relevant endpoints that you have, are matching or there's available information, I think then your change is a much more simpler and may not even require additional extensive testing. Maybe you should still run like a cytotoxicity test and, look at some other specific things that might be really important for your specific device, but there is a chance to use a scientific rationale to show that you are changing a like to like. Obviously, you need to still have a risk assessment written. But the point is that you need to understand the basic chemistry of your material. You need to make sure that you know what plasticizers are there. You need to understand that really when you're talking about a plastic, that does not just include that plastic. It has plasticizers, it has additives, it could have plasticizers. It definitely has additives in there, and it could also have colorants, and all of these in combination could change the biocompatibility profile for that material. So know what you're dealing with, know your child, right? And as I said, when you have a change, always, always, always make a risk assessment to define what you already have and what additional data, either from the source or for root testing, you really need. And then, as I said, when equivalence for certain contact types or durations can be shown, then additional testing may not be necessary, but that has to be really covered in that risk assessment document. Now, to wrap this thing up, I want to leave you with some key take-home messages. First of all, really, the point is to understand the risks of your device. Know your child. Know what is really important for your device based on what you know about it. A written plan is the first step for biocompatibility assessment. You need to have this plan that talks about your device, the picture book or the scrapbook for your child, the device, how it's used, what type of risks does it carry, what type of information do we already have, and then what we have to cover, what we need to do extra to cover the risks. Biocompatibility should be a design input from the start, so we need to make sure that we think about it as a critical step when we choose our materials. And all material changes must be evaluated, so choose your materials wisely. And remember, the main goal here is patient safety. That is what we strive for. We want to make sure that when you have your final finished device, it is not going to inadvertently cause harm to the patient. So I want to thank you for listening. You can always reach out to me through my email, and you should also go check out that Sotera Academy site, where you have lots of different webinars and white papers where you can learn a lot, not just about biocompatibility, but also packaging, sterilization, and so much more. So again, thank you for listening, and I will now hand over the mic to have any closing remarks. Thank you.
Thank you, Helen. We appreciate that presentation and for your insights on all of this biocompatibility content. We'd like to thank you, the viewer, for attending this session. We hope that you found it to be a valuable experience. Again, please check out any more of our webinars on the Nelson Labs website or on the Sotera Health Academy. And we hope that you have a good rest of your day.